Hello everyone, my name is Daniela, and I'm currently a PhD student in the Laboratory of Comparative Physiology of Pigmentation, Institute of Biosciences at the University of São Paulo. Today, I will present an abstract entitled Temperature Effect on the Molt-cycle of Callinectes sapidus, RATHBUN, 1869, BRACHYURA PORTUNIDAE, as part of the results obtained in my PhD project. The molt-cycle is a physiological and behavioral process that leads to the replacement of the animal's exoskeleton. For crustaceans, in general, the molt-cycle comprises 5 stages, which are pre-molt, ecdysis, recent and late post-molt, and intermolt. The crustacean molt-cycle is regulated by 2 major hormones. The hormones that act positively on the molting cycle are derived from cholesterol, and its conversion into the ecdysteroid precursors begins at the Y organ through several enzymatic reactions. Finally, the Y organ releases the ecdysteroids into peripheral tissues like gonads, hepatopancreas, epidermis, and gills, where they will be converted into active ecdysteroids, ready to act on their receptors. As its name suggests, the molt-inhibitor hormone, MIH, is responsible to inhibit the ecdysteroid synthesis and release, which consequently inhibits the molt-cycle process. The MIH is a neuropeptide hormone synthesizing the crab's neuroendocrine systems that comprise the X organ and the sinus gland in the crab's eye stalk. These 2 hormones are released in antiphase along the molt-cycle. Other important factors that can act the molt-cycle process are the environment factors, represented here as the photoperiod and temperature. Both factors can change the period or the frequency or also the increment. Based on that, our main goal in this work was to evaluate the expression of molt-cycle-related genes, the ecdysteroids levels, and morphological changes in the hepatopancreas of the C-seps crabs submitted to 3 different temperatures: 24 Celsius as a cold control group, 19 Celsius as a cold challenge, and 29 Celsius a heat challenge, under a constant photoperiod regimen of 12 hours light, 12 hours dark. Only male crabs with ecdysteroids levels higher than 100 nanograms per milliliter were selected for experiments. A leaf sample of each individual was collected previous to submitting the animals to any experimental condition. 5 days after transferring the crabs to each temperature, the lymph was collected again, individuals were euthanized, and the tissues were collected. The photoperiod regimen was kept constant as 12 hours light, 12 hours dark. The final ecdysteroids values were subtracted from the initial values for each individual to obtain a delta ecdysteroid concentration for each crab. Our results show that crabs submitted to 19 Celsius presented a reduction in ecdysteroids levels in comparison to crabs maintained at 24 Celsius, 29 Celsius. There were no difference between the control and crabs submitted to 29 Celsius. The MIH precursor gene expression showed a reduction in animals submitted to 19 Celsius in comparison to the other temperatures. Cas-MIH gene expression did not differ among the temperatures. The MIH receptor transcripts in the Y organ did not differ between temperatures, but a reduction was visible in animals submitted to 19 Celsius. The crabs submitted to 3 different temperatures showed remarkable morphological changes in the hepatopancreas. Animals of the control group showed a higher number of B cells followed by F cells. Individuals exposed to cold temperatures showed equivalent numbers of F and B cells, while the crabs exposed to higher temperatures showed mostly R and E cells. Based on our results, the MIH signaling was reduced at the lower temperature, in addition to a reduction of the circulating ecdysteroids. The morphological data indicated that the hepatopancreas is affected by different temperatures, probably due to the change in the metabolic demands of the animals. It's all. I would like to thank my colleagues from the Laboratory of Comparative Physiology of Pigmentation and the funding agencies PAPESP, CNPq, and CAPS.